Hello, hi, a very happy good evening. So this is Krishna Ma'am. We're talking about the story of life on earth, and that's biology. So thank you so much for joining my session today. So we are here towards the end of the lecture, I would say, of photosynthesis, but let's see how much we can finish. So I have to tell you about the C2 cycle, the CAM pathway, the factors affecting photosynthesis. So yeah, so here we are in the last lecture. Yes, W, a very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. And I hope you guys are understand what I'm teaching, and it's not that I'm monotonously teaching. So if you have any doubts based on the topic that I'm teaching you, so please let me know. All right? Yes? Okay. So photosynthesis in higher plants. So let's move forward. So what are the topics that we're going to talk for today? So we are done with so much of topics. So we still have C4 pathway left. So then we have the C2 pathway as well today. So we're going to talk about C4. Okay, I'm sorry, I forgot about the C4 pathway is left. So we'll talk about the C4 pathway, the C2 pathway, the CAM pathway, the law of limiting factors if time permits, otherwise I'm not rushing, and then we'll talk about factors affecting photosynthesis. So your C4 and your C2 pathway are pretty important, so please pay good attention to that. Okay, so let's move forward. So before I move, so I'll give you a recap of what the C3 cycle is. So we know in light reaction, so photosynthesis needs light, and in the presence of light, so you have two types of reaction that is happening inside the chloroplast. So one is light dependent and one is light independent, or the dark reaction. So for the light dependent reaction, so you need light, and the end products are your oxygen, ATP and NADP. Okay. And for the dark reaction, actually, so these are being utilized. So here you have to produce your glucose molecule, right? So we saw how your ATP and NADPH are produced. They are produced by your 𝐾𝐕-osmotic hypothesis, and I guess we know pretty good enough information about that. Now, where are this ATP and NADPH used? And that's when we had Melvin Calvin coming into picture, and he said carbon dioxide is getting fixed. So based on that, we have two types of cycle. When carbon dioxide is getting fixed and the first stable compound formed is a 3-carbon compound, it is your C3 cycle. If the first stable compound that is getting fixed is your 4-carbon compound, that is your C4 cycle. Alright, so these were the two cycles. So we went on to see how your C3 cycle happened. So I also hope you guys remember the primary acceptor, the amount of ATP that is utilized, the amount of NADPH that is utilized, right? So let me give you a quick recap about it. So we are here talking about the C3 cycle. So C3 cycle happens in all plants, irrespective of whether the first compound is C4 or C3. So it happens in all plants. So here your primary carbon dioxide acceptor is RUBP, ribulose bisphosphate, which is a 5-carbon compound. So which accepts carbon dioxide, and now it is getting converted into phosphoglyceraldehyde, which is a 3-carbon compound. So since the first stable product is a 3-carbon compound, it is a C3 cycle. Alright, so here the enzyme is Rubisco, and this is your first step, and this step is called as carboxylation. So the next step, your phosphoglyceraldehyde, which is your 3-carbon compound, it is getting reduced into your glucose or your triose molecule. So this step is called as your reduction step. And for your reduction step, so we need 2 ATP plus 2 NADPH. Fine. So the third step here is your regeneration of RUBP. So RUBP has to be regenerated. So for this we require 1 molecule of ATP. So this is your entire Calvin cycle. So for 1 molecule of carbon dioxide to get fixed, C3 is in higher plants, C4 is in a few plants, so C2 plants. Okay. So most of the C3 plants can have C2 cycle. So plants that are having C3 cycle only can have a tendency of becoming a C2 plant. We will see how. But C4 plants can never have the C2 cycle. We'll also see how. Okay, so C4 cycle is mostly in your dry regions where the plants get only limited amount of water. All right, W? Yes? Fine. So what is the total ATP requirement here? So for one molecule of carbon dioxide, you have totally 3 molecules of ATP plus 2 molecules of NADPH. So, but in photosynthesis, we have 6 molecules. So, the total ATP count is 18 ATP plus 12 NADPH. So, this is your total count in case of your Calvin cycle. So, now we move on to your C3 cycle. So, your Calvin cycle happens in the mesophyll cells of the leaf. Fine? Yes? Okay. So, in only C3 plants, it happens in your mesophyll cells. So, now we go to the C4 plants. So, C4 pathway is also called as your Hatch-Slack pathway. So, that is another name. So, primary carbon dioxide acceptor is a 3-carbon molecule here. So, that is your phosphoenol pyruvate. So, this is present in your mesophyll cells. Okay. So, in case of C4 plants, there is a special type of anatomy that is called as Kranz anatomy, right? So, this Kranz anatomy ensures that there is a lot of biomass and the plant has maximum photosynthesis, and they also have what is called as a bundle sheath cells. So, what is a bundle sheath cell? So, if you are able to recollect anatomy, so you have your in the leaf, so sometimes the vascular bundles are surrounded by bundle sheath cells, especially your monocot leaf, so that is your isobilateral leaf. Okay, so these are present in the mesophyll cells. So the enzyme PEPcase or PEP carboxylase is used here. So mesophills lack Rubisco enzyme here. Fine? Yes? So your C4, because the first stable compound formed is your oxaloacetic acid. Okay, we'll see how it happens. Bundle sheath cells are rich in Rubisco, but they lack PEPcase here. So this is your C4 pathway. So wherever is written here, so it is written, whatever is here, so that it is written in the form of words and statements in the previous slide. So this is your C4 pathway. If you see, I told you C3 pathway only happens in your mesophyll cells. So the work is half easier, you don't have to complicate things. But if you look here, C4 pathway happens in mesophyll cells as well as bundle sheath cells, okay. So here this is the anatomy that it has. So you should understand C4 plants have a special type of anatomy. So that is called as your Kranz anatomy. So Kranz anatomy indicates that they have a large amount of bundle sheath cells. They have increased biomass production, meaning they produce more amount of glucose. Biomass is your organic matter. So they have limited amount of water. So your example of C4 plants can be maize, can be sorghum. Fine? Okay. So here you have the primary acceptor. So it happens in 2 areas. First is the mesophyll, and then you have the bundle sheath. So in mesophyll, what happens? So your carbon dioxide is accepted by phosphoenol pyruvate. So this is your 3-carbon compound, and this is converted into oxaloacetic acid, which is a 4-carbon compound. So that is why this is known as your C4 cycle. Alright? Fine. So here the enzyme is PEPcase or PEP carboxylase. You can say it anyways. So this step is your fixation. So now this oxaloacetic acid is reversibly converted into malic acid. So this malic acid is also a 4-carbon compound. So now this malic acid travels into the bundle sheath cells. So this malic acid, when it comes into the bundle sheath cells, it gets decarboxylated. So this step is decarboxylation because I have the removal of a carbon dioxide. So malic acid is a 4-carbon compound. So it is broken down into phosphoglyceraldehyde, which is a 3-carbon compound plus carbon dioxide. So now this carbon dioxide enters the C3 cycle. So this is the reason I said all living plants have your C3 cycle happening. So even C4 plants have your C3 cycle happening. Now your PGA will be transferred back into the mesophils and for the regeneration of your PEP. So this helps in the regeneration of your PEP bag. So this is how your C4 cycle happens. So it happens in 2 particular locations. One is mesophil, the other is your bundle sheath cells. Alright, clear? Yes? Understood? So we know in C3 cycle, we already have 3 ATP for 1 molecule of carbon dioxide. So we have 3 ATP plus we have 2 NADPH. Okay, yes, PEP is 5 carbon, oxaloacetic is 4 carbon. How you remember this thing? No, PEP is 3 carbon compound. Okay, so you have only 2 small cycles, so it is not really tough. So repeated reading and repeatedly teaching, so it is always etched in my memory, so I don't think I can ever forget it till I die. So yeah, RUPP is 5 carbon compounds. So I remember it as and when I studied 11. So it is still not gone out of my memory. So when I started teaching, it was all the more clear. So yeah, so C4 cycle, the product is oxaloacetic acid. So it is 4 carbon. So phosphoglyceraldehyde is 3 carbon. So that is why it is a C3 cycle, right? So RUBP and PEP case you have to remember. So RUBP is 5. So PEP is 3 carbon, right? So yeah, you have only 4-5 terms, so I don't think it is tough to remember. So remember PEP, so only 3 words, so it is 3 carbon. So oxaloacetic acid, so that is very easy. So RUBP, so that looks a little big, so remember it as 5 carbon, alright? Okay, so in C3 cycle, we know 1 molecule of carbon dioxide gives you 3 ATP plus 2 NADPH, right? So in your C4 cycle, you have extra 2 ATP. So totally C4 cycle gives you 5 ATP plus 2 molecules of NADH. But this is for 1 molecule of carbon dioxide. So for 6 molecules of carbon dioxide in C4 plants, you have 30 ATP plus 12 NADPH. Fine. Is your C3 cycle clear? Yes. Sorry, C4 cycle clear? Any doubt? So can we go to photorespiration? So can we just talk about photorespiration today and wind up? So CAM plans and the factors I will teach in the next class. So as of now, remember, is it clear? Yes, any doubts so far? No? Everything is clear? Ask me if you guys have any doubt. So what is the difference, main difference between the C3 and C4 plants apart from the stable product? Yes, I agree, in C3 plant, the stable product is your phosphoglyceric acid, and in C4 plant it is oxaloacetic acid. But apart from that, you have a difference, that is photorespiration. So we are going to talk about what is this photorespiration. So in the first step of Calvin cycle, carbon dioxide fixation step happens, right? So RUBP combines with carbon dioxide to form your glyceraldehyde 3-phosphate. So it is catalyzed by Rubisco molecule. So Rubisco is the most abundant enzyme in the world, but it can also have the active site for both carbon dioxide and oxygen. So in your C3 cycle, the primary acceptor is RUBP. So this accepts carbon dioxide in the presence of Rubisco. And it forms your glyceraldehyde glyceraldehyde phosphate, which is a 3-carbon, and this is a 5-carbon compound, right? So this Rubisco is the most abundant enzyme in the plant community. So this Rubisco, so it has active site for both carbon dioxide and oxygen, because the full form of Rubisco is RUBP carboxylase as well as oxygenase. So it can bind to both carbon dioxide as well as oxygen. Yes, ma'am, a very happy good evening. So thank you so much for joining my session. So please do like, share and subscribe. So it can bind to both. So under what condition it will bind to carbon dioxide and under what condition it will bind to oxygen? Rubisco has greater affinity for carbon dioxide when carbon dioxide is to oxygen is equal. So when the concentration of carbon dioxide is equal to the concentration of oxygen, then your Rubisco binds with carbon dioxide. If this is not equal, it binds to oxygen. Okay, so what if it is so, the binding is competitive and if the relative concentration of carbon dioxide and oxygen determines which of the two will bind? So we can actually directly go to the picture. Yeah, I have the picture here. So here what happens, the carbon dioxide is to oxygen concentration is not equal. So your RUBP now combines with oxygen and instead of forming 3-phosphoglycerate, it forms phosphoglycolate plus PGA. So PGA is formed anyway, but it also forms your phosphoglycolate. So this phosphoglycolate is a 2-carbon molecule. Okay, so what it says? So here it forms 1 molecule of phosphoglycerate, PGA, and phosphoglycolate in a pathway. So it forms this extra 2-carbon compound also. But it does not lead to the formation of any sugar or ATP. It simply utilizes ATP and releases your carbon dioxide. That is the worst part. So there is no synthesis of ATP or NADPH. So C4 plants, photorespiration does not occur because in C4 plants there is internal production of carbon dioxide, because inside the bundle sheath of C4 plants you have the production of carbon dioxide which undergoes your C3 cycle, right? So there is no question of C2 cycle occurring in a C4 plant. Only in C3 plants we have a huge problem. They have a mechanism that increases the concentration of carbon dioxide at the enzyme site. It takes place when the C4 acid from the mesophyll, that is the malic acid, when it comes to the bundle sheath cells, it is broken into carbon dioxide plus PGA. So this results in increasing the intracellular carbon dioxide concentration. Thus only carbon dioxide binds. So if you see, your photorespiration happens in 3 locations: chloroplast, peroxisome and mitochondria. So this phosphoglycolate will now be converted into glyoxylate inside the peroxisome, glycine, and then this glycine will be passed into the mitochondria where it will be further converted into serine and then it will go back as glycerate. So this is your C2 cycle. You see there is no production of any type of sugar, stable sugar here, and there is no synthesis of ATP. All are amino acids. Glycerate, serine, glycine, they're all amino acids. They're not sugars, right? So it's a complete waste, I would say. Fine? Yes. So we'll stop here for today. Why C4 plants photorespiration does not occur? So which means you haven't understood. Wait, I'll explain. So W, remember, in C3 plants, okay, so RUBP is the primary acceptor. It accepts carbon dioxide. So, but the enzyme here is Rubisco. So, Rubisco has active site for both carbon dioxide as well as oxygen, right? Okay. So, when will carbon dioxide bind? When the concentration of carbon dioxide is to oxygen is equal, carbon dioxide will bind. And it will give you phosphoglyceraldehyde, that is your 3-carbon compound, right? So this is your first step in your C3 cycle. Suppose if this concentration is not equal, then oxygen will bind to the active site of Rubisco. So if oxygen binds to the active site of Rubisco, along with the substrate RUBP, then it leads to the formation of a 2-carbon compound, that is phosphoglycolate. So this formation of a 2-carbon compound is a waste of time because it does not produce any sugars or neither it produces any type of energy molecule. So the plant does not benefit from this, okay. But why is this condition happening? Because of the less concentration of carbon dioxide. The balance is not equal. In C4 plants, if you remember, the primary acceptor is PEP. So here I have PEP case and the compound formed is oxaloacetic acid, which is a 4-carbon compound which is irreversibly converted into malic acid, which is again a 4C compound. This enters inside your bundle sheath cells and then it undergoes decarboxylation where it produces a phosphoglyceraldehyde plus carbon dioxide. So in the C4 plant, the carbon dioxide concentration not only depends on the atmosphere. In your C3 plant, this carbon dioxide comes from the atmosphere. So that is why if it is low, it has to face the waste. But C4 plants are wise enough because they have their own carbon dioxide production here, which in turn undergoes your C3 cycle. And this helps in regeneration. So thus, there is no possibility of your C2 cycle happening in your C4 plant. Understood W now? Yes? Clear? Any doubt? W, did you understand now? So far clear? So C3 cycle, C4 cycle, C2 cycle clear. What happened W? Still you are confused? So when the carbon dioxide concentration is not equal to oxygen, photorespiration occurs, that is your C2 cycle, okay. In C3 plants, the carbon dioxide comes only from the atmosphere. But in C4 plants, this carbon dioxide comes from the atmosphere, but there is also another synthesis of carbon dioxide which is internal. So there is enough amount of carbon dioxide always. So a C4 plant is safe, so C2 cycle does not happen. It is as simple as that. Understood? Yes? Alright, so we'll stop here. So we'll continue with CAM plans and the factors affecting on Monday. So please have a great weekend. I'll meet you on Monday. So until then, this is me signing off. So Monday 4:30. So please have a great evening. So this is me signing off. So take care and bye. Thank you.